My name's Tom. I'm one of the BrisSynBio fellows that was hired last year. And I work specifically in the field of synthetic biology. And I'm interested in kind of reprogramming how microbes behave. So I come from a more computer science background, but I've kind of diverted into kind of more engineering and now biology. And today, what I wanted to do was kind of talk about something I often don't get to talk about, which is quite nice. And that's this kind of intersection of how automation is being used within synthetic biology and biology more widely. And I'm really going to basically bombard you a little bit with some of the what I think personally are really interesting technologies that are arising within this field and how they're being used, and not just how they're used to maybe do something more easily, but also the new types of science it potentially opens up to the field as a whole. So to give you kind of a brief outline of what I wanted to sort of like go over, I wanted to start by trying to highlight some of the challenges I personally think synthetic biology faces, which we can't sort of get around at the moment. And then try to sort of like give you an overview of some of the technologies that are related to automation that might actually be able to help. So not just, I said here, not just about getting more stuff done but how we can potentially use automation to start enabling us to handle uncertainty in a slightly better way, ways that it can potentially democratize biotechnology across the world, and also potentially address some of the issues we have in science around reproducibility, which is really important if we want a solid foundation on which to build stuff on. And then in the end, I just want to touch on kind of one of the areas that I think is really interesting in automation that sometimes doesn't get so much press in the news and things like that. So this is kind of an outline of how I wanted to sort of structure this talk. So to begin with, I wanted to kind of go over what I personally think are some of the real big challenges that we face as synthetic biologists. And so I think one of the main things is that I'm an engineer. I'm trying to apply engineering principles to biological systems, but biology is really messy. Now that isn't really a very quantitative, doesn't have really a quantitative meaning, but what I personally mean by that is the fact that unlike, say, a building that might have an overall plan for how everything is pieced together, and there's some kind of underlying sense that kind of we can understand how it's kind of designed, biology evolves. So often you find that you have to really understand its history to understand why it's structured in a particular way at this point in time. And that means that to us that come from a different perspective, it can look very messy and very complicated and very esoteric and have all these weird like things going on that we don't really understand. And so this makes engineering it very difficult, because often we want to make use of existing biological parts in new ways. And so if we don't fully understand how they've evolved to perform that function, we may not fully understand how to piece them together in the most effective way. Another really important part and a really important challenge, I think, in synthetic biology is this idea of that these are complex systems that are composed of many interacting parts. So if you look at even the lowest level, like molecules within a cell, you have huge numbers of these things that are colliding and carrying out various chemical reactions. But at all levels, even up to the level of entire organisms that interact with an ecosystem, there are a huge number of interacting parts which actually makes these biological systems tick. And so this is a real problem for engineers, because as an engineer, we're very good at building things in isolation. So we often, if you think, I think probably the most obvious example is when you have your aerial, or you used to have your aerial that you plug into your TV, you have shielding to insulate what's going through that wire from everything else that's going on in the world. And that's quite difficult in a biological context, because inherently a lot of the functions that biology displays require these interconnections for them to actually work effectively. And so this makes it very difficult for us to potentially take parts from different organisms and piece them together in new ways, because there are often contextual effects that we haven't foreseen or we don't necessarily know, which make that very difficult or mean that they behave in an unpredictable way. The final part that I want to highlight is that we have quite a limited knowledge of biology as a whole. There's a huge amount of things going on that we don't always understand how they work. Even so, we'd still like to make use of some of these processes or components that are there. And so we have to be able to deal with this kind of high levels of uncertainty in how these actual things function. And this is very different to, say, for example, civil engineering, where the actual components we're building with, we can actually characterize in a pretty complete sense. We can understand how strong a piece of stone is, or how malleable like a piece of rubber is. And it's very difficult in a biological setting to do that because we don't necessarily know all the things that we have to measure to actually fully quantify how it actually works. So I think all of these aspects are really difficult to handle, but I hope by showing you some of the technologies that people are starting to use, you can start to see how automation can potentially give us a way of tackling or at least trying to alleviate some of these problems in kind of an interesting way. So to sort of like begin this journey, I guess, I wanted to start with kind of one of the foundational technologies that we kind of rely on in synthetic biology, and that's DNA synthesis. So this is really one of the kind of cornerstones of what allows us in synthetic biology to actually do some interesting things. So DNA obviously is what they call the code of life. But what's kind of interesting is that when we want to actually write our own pieces of DNA or write our own biological programs, the chemistry we use for producing those synthetic pieces of DNA is actually it's fairly basic in some sense. So it's basically this kind of cyclic, sort of sequential process where we add an individual base at a time and we produce these kind of small lengths of DNA or single-stranded DNA called oligos that we can then piece together to make longer pieces of DNA. Now, one of the reasons that kind of synthetic biology I think has really taken off over the last kind of decade is that when you look at the productivity across the world in terms of how much DNA we can actually write, it's increased in a similar way to how kind of transistors might have increased in microprocessors. So it follows this kind of what's known as like a Moore's law, or an exponential growth effective in terms of how much DNA we can actually synthesize. And although the process itself is quite simple and basic and sequential, which isn't ideal for making big pieces of DNA, a lot of these improvements have come directly through just automating that process using machines rather than humans to actually carry out the synthesis process itself. One of the interesting things that's happening more recently is that although you can automate some of these processes, ultimately they're still going to be very expensive if you're using large amounts of reagents or products, and you're going to produce a large amount of waste if you're doing it at large volumes. And so one of the really interesting areas that one specific company called Twist Biosystems has been pursuing is to actually scale down, not just automate, but scale down the process to the point where you can, rather than performing one reaction to produce one piece of DNA, you can actually perform 1,000 using the same volume of actual reagents. And that has a really big knock-on effect because it not only reduces how much you're using, but it also reduces the cost significantly. And there's a sweet spot in many of these systems whereby if the cost gets cheap enough, people can actually start using this in an effective way to produce interesting biological systems. And so this is kind of an interesting way of combining not just automation but miniaturization to allow us to actually tackle some interesting problems in DNA synthesis. Now, being able to make lots of DNA doesn't actually sound that interesting. It's not really that interesting in many ways, but one of the things it can potentially do is actually provide us with an alternative way of actually tackling difficult biological problems. So historically, if we were trying to build a synthetic biological organism, we might have an idea of how we're going to piece together various bits and have an idea of what the DNA might actually look like. But there might be some options that we have with some uncertainty into how exactly we should piece those things together. And what we'd probably try and do is argue with ourselves about what's the optimal way of piecing these things together, because it would cost too much to make every possible design that we would want. And so we'd come up with a few that we think really should work or would test a particular hypothesis we're interested in about what is going to actually help this system work. And we'd then synthesize these and try and from that gain a better understanding as the mechanisms of how this system works so that we can predictively design new systems in a better way. If we can synthesize huge amounts of DNA, then there's actually one of the approaches is that we just, rather than think too carefully about every little detail, we kind of appreciate that we do have a lot of uncertainty in the design itself. And we say, well, actually, we don't know what the best way of arranging this genetic circuit is. So we'll just build 10,000 of them and see what happens, and just use this very much data-centric view on science in some way. So this kind of moves away in some sense from kind of a hypothesis-driven science more to try it and then use these kind of statistical models to try and infer what interesting relationships there are in the data itself. And so sort of like in this example, the idea would be you kind of build a huge number of these things, and then you could start to use what's often called black box methods, such as deep learning or kind of standard statistical methods, to try and find subtle relationships between these different designs to figure out what might be the important features that are necessary for the function that you want the system to perform. Now, this approach is a very powerful approach if you have enough data. So there is a threshold over which you need to get before these approaches actually work. And it's kind of why you've seen this explosion in machine learning and AI in computing, is that basically these companies have collected enough data that they can actually start seeing interesting patterns in some of these systems. But one of the big problems with these systems is that they move away from really a mechanistic understanding of the system itself, which makes it, from an engineering perspective, more difficult to maybe predict how something that's never been built before might actually behave. So there's this kind of trade-off, and I think kind of a conflict in some sense as to which approach might be better or worse in certain situations. And there are some interesting work as well around using these kind of black box methods on themselves to try and infer certain mechanistic understanding from what these systems have actually found out. So I think this kind of highlights a really interesting way that through automating a very basic process such as DNA synthesis, we can actually do science in a different way that we wouldn't have been able to do before. So a lot of what I showed in the previous example really related to a scaling down of the volumes that you're using so that the costs could become kind of negligible in some sense. And another way that that's being done is through a technology called microfluidics. So this is where, rather than, say, doing chemical reactions in big tubes, you basically shrink everything down. So on the size of kind of a small microscope chip, which is kind of what one of these things is, you have these little channels which carry, is this going to work? Good. You have these little channels and valves and various, effectively, like canals, I guess, for fluids to flow through and react in different ways. And what this allows you to do is to shrink a big chemical process down to a very, very small scale so that you can effectively replicate it a large number of times on a single chip. And so carry out a far larger number of experiments than you would have been able to previously. Now, one of the things that's nice about this technology is that we can exploit a lot of the existing mature technologies for actually producing things such as chips at a very low cost. So a lot of the technology behind this is based on kind of photolithography, which is used when we actually fabricate microprocessors, but it can be repurposed to be used in a biological setting. And so I think there's also going to be a lot of these kind of reusing existing technologies in new ways that are relevant to biology, which I think could potentially be really, really interesting. One of the major limitations of this type of approach is that the design of this chip is kind of fixed. So it has a single purpose that it has to fulfill in some sense, and it's not very adaptable. So if a process changes in some way, you have to basically redesign the chip from scratch. One of the really interesting new technologies that's also starting to come out is something called digital microfluidics, where rather than having a fixed geometry or structure for your chip, you instead have these kind of arrays of electrodes that can basically pull small discrete amounts of fluids around, and by moving them around you can mix different ones together, you can have regions that are heated or cooled. And what's beautiful about this sort of technology is that you're not defining the process by the chip itself, you're defining it by how you move these things around. So it means that this type of chip can be used for a whole range of different processes you might want to actually test. And so that makes it incredibly flexible and potentially adaptable for new things you might want to do, and allows you to focus on, if you can make a really good one of these, you can then open up automating a really wide range of different processes that you may not have thought of doing before. So this scaling down of chemistry to kind of microscopic levels can have a real big impact on how we understand some biology as a whole. And so one of the things I spoke about at the start is the fact that biology is very interconnected. So if you look at, say, an organ in your body, it's not that every cell is exactly the same. Some of them behave differently and they have different features and they may have different functions in some sense. But what's been done previously is that if you took, say, an organ like your brain and you want to understand how the cells are behaving, you would effectively mush them up and then get an average view of what's actually happening within those cells. Now that obviously isn't going to give us the information we need to know about how the system as a whole is actually working, because actually it's composed of many different cells that are probably quite different in the way they're behaving or the state that they're in. And another example, I guess, is also the microbiome, which consists of a huge number of different species of bacteria and microbes, which all are different in some sense. So what we'd ideally like to do is to be able to look at each of these individual cells in isolation. And so one of the interesting ways that we can do that is to take some of these microfluidic systems and use them to actually encapsulate the individual cells within these kind of tissues or multicellular systems and carry out chemistry on each of those cells individually. So the way that this is done is using what's known as like a droplet-based system normally, or it's one of the ways it can be done anyway. And so the idea is that you have some kind of liquid here that might contain a mixture of the cells that you're interested in. And it will basically, if the density of the cells is low enough, then when these droplets are being produced, you'll end up with just single cells being isolated in each of these droplets. Now, the thing is, when you look at tissues and things like that, the numbers of cells that are involved it's millions and billions of cells. So you need to have these things highly automated and highly fast in order to get an actually useful representation of what's actually going on. And so one of the things that microfluidics can really allow us to do is to start to basically scale up some of the analysis we'd like to do of the chemistry we'd like to do, so that we can actually start better understanding some of these really complex, say for example, tissues or multicellular systems that are present everywhere in nature. So that was kind of an example of how microfluidics is being used. Another aspect of biology that synthetic biologists are also interested in trying to exploit is evolution. And I think probably one of the most famous evolutionary experiments was carried out by this guy called Richard Lenski, who basically started off with, I think it was 12 identical bacteria, and he placed them in media which they're not normally grown in. So they weren't really adapted to living in that environment. And what he's done, not just himself, but a lot of PhD and postdocs have done, is basically grow those cells up and dilute them every day for basically 25 years, or I think it's more now, I can't remember, probably it might even be 30 years now. It's like a huge length of time. And the idea was to try and understand, or basically have a way of actually observing evolution in process. So through as the cells grow and divide, there'll be mutations that get taken up in those cells. And as those mutations, if they potentially have a beneficial effect on growth, then they'll get fixed in the population. And you'll start to see actually evolution of the phenotype of those cells over time. Now, this is obviously like a huge experiment. It costs, I don't know what it costs, I think there it's got some number, like $4 million. But that probably doesn't count all of them, probably doesn't count the man hours and things as well. So it's like 75 person years of actual effort. So this is kind of like crazy to think, you wouldn't get funding to do something like this now. But one of the things that this kind of, I think, inspired in a lot of ways are these synthetic biology kind of systems that try to kind of automate this process of growing cells and then diluting them, continually growing them, and using kind of natural evolutionary processes to produce something useful. And one, I think, really good example of that is something called MAGE. So this is a system that came out of George Church's lab at Harvard. And whenever he speaks about this, he'll always call it it's basically a way of accelerating evolution. So the idea is that rather than having people effectively doing what would normally be done, he has this kind of, it can potentially be done with people, but it's much easier if you have some kind of array of robots. So these are kind of different plates that these cells might be grown in. These are kind of robots that will dispense different things. You have different areas here that will heat them up or cool them down, and all sorts of other bits and bobs. And the idea is that you can use this kind of natural evolutionary process, but you can also introduce small pieces of DNA that you kind of force the cells to uptake, such that when they're replicating their genome, you can bias where mutations in that genome actually occur. And this allows you to then basically kind of evolve them towards some interesting goal very, very quickly. So you can run these machines for months. I think some of these experiments, they ran it for 3 to 6 months to actually get improved phenotypes. So this is kind of an overview of the process. You have all these pumps and you have various cells, and it goes round and round in a cycle. But by using this and biasing the way evolution works, they've shown that you can basically make modifications to genes that are involved in, for example, a specific metabolic pathway that maybe produces a chemical we're interested in. And the modifications that get taken on, as long as they have some beneficial effect on growth, then those cells will basically become more highly propagated, and you end up fixing certain mutations that lead to the optimal production of some product that you're interested in. Another really interesting use of this process is that there's quite a lot of interest in potentially recoding organisms. So bacteria, or all life, uses sort of a certain alphabet of amino acids that code for the various pieces that make up proteins. And one of the things that they've managed to be able to do is to take one of those letters effectively and remove it, so it's not needed by the natural host. So they've kind of recoded how that organism works. And what that does is it frees up basically a letter in the alphabet for them to maybe introduce artificial amino acids or other types of things they might want to incorporate into proteins. And additionally, because that amino acid might be essential for other organisms such as viruses, it can also make them immune to certain viruses. So there's a lot of being able to kind of recode organisms through these evolutionary processes is a really powerful way of potentially modifying their phenotype in some sense. And it's really led to a large number of different breakthroughs in that area. Another technology that I'm sure probably a lot of you are aware of is obviously 3D printing. Now this is obviously automating the production of some interesting geometrical shape that you want to produce. I think it's like a dragon in this case. And what's nice about this technology is it's kind of a way of moving from kind of producing things in big factories and centralized places and allowing kind of anyone to kind of produce something that they want locally. So kind of basically making it available to everyone. So more distributed manufacturing in some sense. Now, one of the nice things about 3D printing and the way that it might fit in with synthetic biology is that it allows us to very specifically place pieces of material in a certain three-dimensional geometry. And as I said earlier about how often biological functions are really reliant on interactions with other cells or things in their vicinity, they're also really reliant on kind of the physical properties that they experience. So for example, when things develop, it's important that they have a certain structure for the cell to know that it's in a certain position and to behave in the correct way. And so one of the ways that these kind of 3D printers are being kind of repurposed is to actually, rather than print, say, a particular type of plastic or use some type of resin, is to actually use a kind of a biological ink or like a hydrogel effectively to allow you to print things that not just have some material, but they also include some actual living cells themselves, and allow those cells to then develop in a more natural way to produce, for example, in this case, it's a kind of a replacement ear, but it potentially could be anything. And from a synthetic biology perspective, what this opens up is it means that we don't necessarily have to fully understand all of the developmental pathways to producing something. As long as we can get the cells in the right place, we can actually get around that limitation. And so this actually simplifies, in some sense, our abilities to potentially make some really complicated multicellular structures that might be really useful for different applications. Okay, so I kind of mentioned there about this idea of distributed manufacturing, which I think is kind of a really powerful kind of technology for many different reasons. But one of the things that I think is really important to highlight is that biology itself is effectively a distributed manufacturing technology. That's what it does. It takes materials in the vicinity and it transforms them into things that it needs to live or like to make structures for itself. And I think one of the really interesting papers that only came out earlier this year was around trying to create almost like a 3D printer for biology, and that's kind of how I see this. And so the idea is that you might have some biological thing that you want produced, and that can be encoded in some kind of digital file that you have. It can then be sent to what they call a digital to biological converter, or DBC, which basically includes a kind of range of different equipment. So this has on it a DNA synthesizer. It has various machines to help with the actual assembly of the DNA into actual pieces of DNA that can be used. And it has other parts which allow that DNA to then be expressed to produce functional biological pieces of material, for example, RNAs, which could be used for vaccines, proteins or viral particles. But you could imagine that this could be expanded to the point where it actually makes a completely new type of cell, which could then develop into something that we actually want. And the really powerful thing here is that it's fully automated. So in a lot of those other systems I showed you, there is some human intervention that's required. Once this thing is set up, actually it will just do all of these processes itself. And that's really powerful because what it potentially allows is for this to be used to generate things very, very far away from here potentially, where would be very expensive to physically transport the materials that you need. And so one of the areas that this group is particularly interested in is how this could potentially be used to allow for the manufacturing of goods at point that it's required. So for example, if we want to explore space, we do not want to have to take everything we need from Earth to another planet because it would be impossible to do that. Instead, it's much better if we can just utilize the materials that are present at the location we're at and convert them into things that we can actually then use and potentially want. And so this type of distributed manufacturing or distributed biology I think is a really powerful paradigm for potentially making that possible where it wouldn't have been previously. Related to that type of idea, there are also a number of startups that are kind of, I think, beginning to kind of become a little bit more well recognized, especially in the US, that are taking that similar idea but not doing it on such an extreme scale. So one of the kind of the most well known, I think, is probably a company called Transcriptic, which is based in California. And their idea is that you can have this box, so this is like I guess like a shipping container, and inside it they have machines that are kind of would cover 95% of what any synthetic biologist might need to do. So it has things like PCR machines, it has different temperature controlled areas, it has various other bits. And what was really critical for them being able to make this possible was the fact that they used, they had to come up with their own automation technology to allow for material from one device to be put into another. So a lot of these machines aren't inherently good at being automated, and so that's really where their kind of interesting aspect came from. And their kind of model is really similar to how sort of cloud computing works now. So rather than a company being required to pay a huge amount of money in investing in capital, such as like machines and space and all the rest of it, they allow these things to be used by anyone for a certain cost of time of use. And so the idea is that for companies that are starting out, it allows them to have access to a huge range of different technologies effectively very cheaply and to scale their production as necessary using this infrastructure rather than their own. And so this kind of gets around these problems of needing kind of huge amounts of equipment and materials yourself to using just what's actually kind of centrally available. And so it really does, I think, democratize how synthetic biology is done. And I can kind of attribute to this in terms of like setting up a lab here. There's a lot of equipment, a lot of money you need to do it. And actually thinking about it, you don't use that equipment 100% of the time. And so this is a really, I think, a powerful way of getting the most out of some of these technologies and machines. The final kind of thing or technology I wanted to kind of like spring on you is focused more on this kind of idea of reproducibility. So I think a couple of years ago there was a study that was done around kind of looking at basically cancer studies and how reproducible the results actually were. And it was really sad because I think they found that only 6 out of the 53, these are landmark papers, these aren't kind of small papers, actually had reproducible results. That doesn't mean that the other 53 weren't correct, it just means that in the study they did, they couldn't reproduce the results exactly as they saw. Now this is a really big problem, which kind of stems a lot of the time from the fact that a lot of life science research is done by people. So everyone has their own way that they pipette something. Even the temperature or the environment the lab's in can affect how reactions take place or how quick they are. And so actually when you report a finding, it's really within the specific context of your lab that it actually works often. And so one of the interesting projects, I think, to sort of try and get around this was started by Eric Clavins at Washington University called Aquarium. And it's kind of it's one of those projects which is really cool and really creepy at the same time. So the idea is that he's not trying to automate the processes that go on in a lab necessarily with robots, but he wants to have a set of basically a set of protocols that are basically stating what should be done by a person, and then using effectively monitoring equipment to see exactly how that person is doing that experiment. And so when you try to reproduce a result, you can actually have all the data about how the original experiment was done, not just what someone's written, but physically what they've actually done. And to sort of give you an idea, there's a video from his website here, which I think is kind of interesting. It is very creepy, but you can see they use kind of eye tracking. So the people that are working in these labs are really basically everything about them is monitored. And so they can really start to see how people are actually working in the lab potentially differently from each other, and why certain results potentially might not be particularly reproducible. So yeah, this is about the eye tracking, but some interesting ones about how even people are standing and doing things. And it's like, I find it really creepy, but it goes to show that actually that there's a lot of information that we find it very difficult to put in a highly, like an explicit form. And I think these sorts of projects really help to kind of figure out what it is we say, what we mean when we say, put this into this, or do this reaction, or that type of thing. And so I think it's a really interesting project, and I think understanding how we do science like this is actually just as important as some of these automation techniques in some sense. So that was kind of going through some of the interesting technologies that I think are out there. But one of the things that I think often is forgotten about when people talk about automation is that automation doesn't just relate to carrying out some physical process over and over again, and that it's just as relevant for design as well, which isn't technically a physical process in some sense. And to try and illustrate this, I wanted to kind of highlight a recent kind of thing that came out in the news. So what IBM have managed to do is to come up with a new way of making microprocessors whereby each the features on the chip are effectively 5 nanometers in size. Now what that means is that they can basically cram 30 billion transistors on the size of your fingernail, which is when you put it in the context of your brain having, say, 100 billion cells roughly, what they're fabricating is actually incredibly complicated and incredibly complex. And while the fabrication is pretty amazing, what I find more amazing is that they fabricate this 30 billion transistors and they actually work, they do something. It's not like, I think most companies could probably fabricate something very, very complicated, but the fact that they can fabricate this and it actually has some function that works reliably, that to me is actually more interesting in some regards. And the reason that that's possible in electrical engineering is that we can't physically comprehend all of the things that are going on between these 30 billion transistors. And so we have to make use of abstractions and also supporting tools which actually help with which actually basically automate certain tasks within this process. And so this idea that you have kind of hierarchies that you can physically think at are really important. And so, for example, like when someone's maybe designing this chip, they wouldn't be looking at the individual transistors, but they'd look at small motifs that maybe have a certain function, and they'd know how the things should be connected together. And they would just rely on automated tools to say how these things should actually be connected in some sense. And so this is a really important, I think, point to make is that automation can assist not just the physical process, but also the design processes themselves. In synthetic biology, one of the ways that's kind of being supported is through standard efforts in producing electronic standards that allow for tools that are developed in the different areas that you might need to do for a synthetic biology project, to allow for data to be exchanged between these various tools and kind of support the automation in some sense. And finally, there has been some efforts to try and apply this type of approach to synthetic biology. So this is a paper that came out early last year, where they've effectively taken an electrical engineering perspective. So they use what's known as a language called Verilog to basically define a function that they want this biological system to exhibit. They then have this automated design process, which is kind of the automation part of this. And what this really relies on is having these little genetic devices, these kind of motifs that are really robust, because that makes this automation possible. And through that, it means that they can just give a computer a function, and it will automatically give them the ATCG, the bases in the DNA that needs to be synthesized to actually implement that. And so this is a really powerful way of moving away from worrying about all these low-level interactions and what's happening to a much higher level language that we can kind of more easily reason about functions that we want, to produce really complicated biological systems that wouldn't be possible if we didn't have some of those design processes automated. So with that, I just wanted to end. And I just wanted to kind of summarize by saying that I think automation isn't just doing more of the same. It actually opens up some new avenues and new approaches to doing certain aspects of science. And critically, it's not limited to just the physical process of making something. I think the design processes themselves are also amenable to these kind of automation technologies, and they're just as important in many respects if we want to build these really complicated systems. And with that, welcome to contact me, and thank you for listening, and let's have a discussion about it, I think.